Great, thank you. Okay, so kicking off our first session, I'm going to hand it over to Klaus Romero.
Okay. Thanks, Laura, and thanks everybody being here with us. And thanks to the panelists who agreed to join us after lunch on a topic about clinical trial simulations. And as my good friend and colleague Michelle Campbell reminds me, the number one rule for these sessions is to not get her to fall asleep. So that's what we're going to try for everybody. Okay, so I'm going to kick us off with a couple of thoughts around the notion of these slides. Okay, so let me start with the introduction to the panel. So I'm just going to go in this order. Frank Jarvix is not only a well-renowned CMO, but he's a CPAT veteran. Back in 2011-ish, something like that, on the polycystic kidney disease effort that you're going to hear about. And we have Kevin, who started at the FDA at the same time I joined CPAT back in 2008-ish, or something like that. He was at the Office of Clinical Pharmacology first, and now he's a transformative force in the Office of Neuroscience at Cedar. Alex, he's an executive director within the Rare Disease Cures Accelerator Data and Analytics Platform, which is the big platform that is now becoming the home for every precious patient-level data points in rare diseases. Ramona is a multi-talented scientist that has played many roles at the organization, but she now leads the Duchenne Consortium, and we're going to hear some pretty important learnings from that effort. And then we have Soren, who was a collaborator in the early days of the PKD Consortium and now has jumped ship and joined us, and now he leads the polycystic kidney disease efforts that also tie in with RDCA-DAP. So with that, let me walk you through a couple of, there we go, aspects of the regulatory science pillars. This is what we heard this morning loud and clear from both the agency and our industry colleagues. Everything boils down to the clinical context, and that is absolutely important. Angela reminded us about forgetting about the last NDA carried forward, or the last paper carried forward approach. This is what we're here to start figuring out. What is the specific context that can apply to multiple diseases, and what context applies specifically to one disease or a family of diseases? If I can get this to work, there will be an animation about the totality of evidence. Let me go back. Or, well, I'll leave it at light. So, totality of evidence. We also heard loud and clear that it's the totality of evidence. There's no one-size-fits-all definition for totality of evidence, but it hinges on the context. The totality of evidence cannot be viewed outside of the clinical context. That's the other piece that is important. And you're going to hear perspectives of how the different data sources and their integration and the transformation of those data sources into quantitative insights have been able to transform some therapeutic areas. But as we know, there will always be some underlying uncertainty that will always be there. And the important thing is not to get scared by the remaining uncertainty, but embrace the fact that through the integration of data and through quantitation, we can put a quantifiable number on what that remaining uncertainty is. And as science evolves, we might be able to generate hypotheses to where that underlying uncertainty comes from and then try to explain it in an effort to continuously shrink the underlying uncertainty. And the last piece is the specific regulatory pathways with the flexibility that we heard from the agency in the right context, with the right totality of evidence, with a quantitative understanding of remaining uncertainty. That's really what drives decision-making for regulatory science, especially in rare diseases. So, what RDCA-DAP brings is the foundation to be able to integrate all those data sources Every data point in rare diseases is absolutely precious, and RDCA-DAP provides a home for those data to then transform those data into specific actionable solutions, some of which you see listed there. But the notion of being able to simulate clinical trials and use that to optimize trial design touches on endpoints, touches on biomarkers that can be used as endpoints but don't necessarily need to be always thought of as endpoints. There's a lot of biomarker use that can be applied to enrich clinical trials, optimize how you select patients for clinical trials, and that's very impactful, and that's not necessarily an endpoint. Of course, the patient-reported outcomes and the different types of clinical outcome assessment tools so that we understand exactly to measure things that are clinically meaningful is absolutely critical. But then everything boils down to the data. And we have Ramona Walls in the audience here, and she's going to help us with some of that as well. So this is a summary of that. It's all about integrating data. It's all about making sense of the data. We heard how each data source speaks both literally and figuratively different languages, how ontologies can help us make sense of the data. We heard about the importance of the efforts that are going on in disease staging, clinical outcome assessment optimization, and the topic at hand, drug development tools that are quantitative in nature and that can help us put all of that together. So, this is what this is about, making sure that we take all the data sources that you see there before the funnel, have the funnel be putting the data to speak the same language, and then making sense of the data for three big categories. Really understand the disease. What are the sources of variability that can help you understand differential populations in terms of their progression rates, and what are the sources of variability? On the other end, the aspects about drug effects and how the drug effects can change the nature of the disease or not. And then the aspects about clinical trial. We talked about placebo effect and how it's important to understand if the placebo effect is an add-on with an onset, a magnitude, and a duration and a variability to just, quote unquote, underlying disease progression. And then the dropouts. And understand what is the time-varying probability and the drivers for patients dropping out of the studies. This is pretty resource-intensive, data-hungry, but we have the track record to be able to do that, and that is what we're going to be discussing today. So, with that, let's kick off the panel. So, Frank,
yes.
you were part of the transformative journey in PKD, and you saw how the integration of data led to some pretty impactful quantitative understanding of not only the disease progression, but the biomarker dynamics. And you were able to benefit from that from an industry perspective. Can you kick us off with some comments on that front?
Sure. Let me just start by saying all of that was way before this. It began really with the Critical Path Initiative that was both listed for FDA and NIH in studying this particular rare disease, autosomal dominant polycystic kidney disease, which sounds very obvious. Your kidneys are normally this size. With that disease, they can grow to be football, rugby ball size, and stop functioning. So the NIH actually had launched a natural history study in patients that were very pre-symptomatic but potentially likely to progress. And those data became available around the time our company was interested, primarily based on animal data showing effects on those animals' kidneys in slowing kidney disease and their kidney growth and thereby slowing the loss of kidney function. We used their data without any sophisticated analytics to help design our first pivotal trial, which, although it was a rare disease, ended up being conducted in 1,450 subjects or participants over 3 years. And it was a challenge. And there were a lot of negotiations and discussions with FDA at the time. There was a tremendous amount of uncertainty with regard to that endpoint of kidney size and what it really meant. But how we used it really was to say, we're going to only treat those patients that are likely to progress to renal failure. And the data that NIH brought forward was very suggestive that there were certain types of patients with a particular type of PKD called ADPKD type 2, or whose kidneys were relatively small for age that didn't really need to receive treatment. So we focused on the patients that were more likely to progress, and that actually did help with their disease. Now, what happened after that, as I said, FDA, NIH came together in a series of meetings, coordinated, I guess, by Critical Path, beginning in 2007, where at the very first meeting, they were like way over here. We really didn't understand what FDA needed. We hadn't been talking to each other. That meeting brought everyone together, and with a lot of support, I would say, from FDA, a lot of high-level people were at each and every one of the meetings and still are part of the consortium at every consortium meeting, really helped bring that endpoint and how to construct a clinical trial properly together. We had already started, and we did modify our trial to some extent on the way. And it was very helpful at the end of the trial when we saw that that particular endpoint was highly positive, but also had some interesting nuances that needed to be analyzed further. So ultimately, that drug received approval in almost every country before the endpoint was approved. And that endpoint was not approved as an endpoint, but really as a clinical drug development tool to enhance or enrich the clinical trials. Ultimately, when the drug was approved with a second trial in the US, or for the US, I should say, it was global. This was a 1,370 patient trial for 1 year. That data combined with the first trial led to FDA's acknowledging or accepting that as a viable or validated surrogate endpoint for approval, which then other companies, I think, benefited from in terms of designing their trials in a more streamlined, cost-effective manner. And the company I was working with at the time, Otsuka, benefited in terms of using it for analyzing or as an endpoint for pediatric trials and other sorts of trials that would potentially be helpful.
And that endpoint was total kidney volume.
Total kidney volume, yes.
Which is an imaging type of endpoint. Again, the notion of how the biomarkers link to the clinically meaningful aspects of disease. I wonder if you can comment for 30 seconds on how the underlying data in the first round had imaging data that came from 3 imaging modalities,
right,
and how we were able to integrate that.
So the CRISP program, which was the NIH-funded natural history study, incorporated historical data, incorporated prospective data in healthy volunteers obtained only by MRI, which was a more sophisticated, more accurate way of doing this. But there was data from a number of centers who have studied PKDs for decades, including ultrasound, including CT, a variety of imaging modalities that were brought together, and part of the validation of the endpoint of total kidney volume included not only what does the volume mean in terms of disease progression, but also does it really matter which imaging modality one would use? And it really didn't. I mean, there were some preferences and some, obviously you want to avoid radiating, radioactive or radiating imaging if you can. And then there were some other caveats as well, because MRI works really well in adults, but when you have pediatrics, it could be very troubling. So their ultrasound, knowing that ultrasound could work, I think was very helpful in terms of designing trials that would be amenable to that patient population.
Right, perfect. Ramona, you are in the midst of the last development stages for some pretty cool quantitative drug development tools for Duchenne that are currently under active review by both FDA and EMA. But that doesn't mean that the industry members of the consortium can't use the tools before they're fully vetted and endorsed by the agencies. Can you comment on that experience?
Sure, yeah. And thank you for outlining this. So yes, to provide an overview, the Duchenne Regulatory Science Consortium has been working towards multiple regulatory solutions and quantitative solutions specifically. Clinical trial simulation tool that was generated by extensive effort of quantitative medicine team, data managers, and all collaborators that convened as part of the consortium to really create knowledge around the disease progression in Duchenne muscular dystrophy. So we were able to build this disease progression model in a very much of a data-driven effort that allowed us to aggregate data from multiple sources. After the publication of the first therapeutic area user guide in 2017, that allowed us to merge data from different sources. So this enhancement of the analytical power of our data is what fundamentally has brought us the ability to first foremost being able to design these disease progression models, because now we have longitudinal data that can tell us better from the years of 4 age and above for most of the endpoints that we've been working on, and 5 years and above for FVC, specifically the forced vital capacity. We can look at trajectories of disease progression in time, which is a very important tool for design of clinical trials. So we've seen that as part of the consortium activities, it has been fundamental for us to really have an interaction with the drug developers in understanding where the gaps and how we could capture these in a quantitative and mathematical model, and really assessing for the velocity endpoint specifically the detail on how to quantify and standardize the language that each of these studies had collected in very different ways, as you can imagine, going from natural history studies to clinical studies and to randomized clinical trials, really assessing the standard language that allow us to then come down to 5 endpoints that are currently used in randomized clinical trials for Duchenne, and really design these models in a way that is efficient for future drug developers that will be able to use this tool freely and publicly available. So before getting there, of course, there is a process of exploratory use of these tools as part of the consortium. So I think it's really important to highlight the importance of early access for those that have been part of the process to these tools to really inform drug development early at the development stage. So we know that during the process of review of these tools, it's certainly claim that we make that these tools are not fully endorsed, but because it's part of the back and forward regulatory feedback and intensive approach of very sophisticated mathematical models, there is at some point a level of maturity where those tools can actually be used in a clinical trials context to really start refining the clinical trial design. So I think the community has shared with us some of these advantages and advancement as potentially being part of the design, but also making it useful earlier.
Yes. So, but I wonder if you can comment a little bit more on the nature of the endpoints that are being modeled. Because we heard from Angela this morning how it does not just go with the inertia of whatever endpoint came before. So I wonder if you can talk about the journey that we took with the regulatory feedback about which endpoints were selected for modeling.
Absolutely. So I think the initial plan was to include 6 endpoints, and including the Brute score, which is a scale that then was considered not as consistent for measurement to be included into these mathematical models, just because, again, this is a data-driven effort. So the granularity of the data is what is driving then the model fitness. And so really the definition of the rigor in which the data were collected is also what has brought us to define that the endpoints selected at the end were 5 and were North Star ambulatory assessment, velocity to climb 4 stairs, velocity to walk around in the 10-meter test, and velocity to stand from a supine position. We also were able to include among the many respiratory tests that are collected in clinical trials and observatory studies, only FVC, because unfortunately we know that it is complex to characterize respiratory testing in a pediatric population. And really, the only endpoint that consistently showed, being collected in a granular way across all the data sets. And we're talking about 15 to 20 data sets that were combined. So it's still a great amount of data, but the granularity is important, and the rigor of standard collection mixed with the standardization of the data next, of how it can be analyzed, it's extremely important for us to then generate solutions that are regulatory ready.
Fantastic. And the number of data sources that you are mentioning in a rare disease is a pretty impressive result.
Yeah, and this is also, just to put another point, I think increasing the data sources is what caused us to decrease the heterogeneity and variability that we have in these data sources. So I think that having these tools of really aggregating data from multiple sources, knowing that there are limitations in, as we've seen, characterizing dropout is not as easy in natural history studies or observational studies as it is in media for clinical trials instead. So there are limitations in looking at multiple sources of data, but certainly depending on the purpose and the context of use, you can find a benefit from doing so.
Perfect. And just to plug that, tomorrow when we do the session about innovative trial designs in rare diseases, we're going to delve deeper into the notion of the underlying variability and underlying uncertainty. But about the data, I'll turn to Alex now. So you are on a mission to make sure that every precious data point is maximized in its utility. Can you comment on that?
I think it's more of the mission of our team than just mine. Speaking as a non-data scientist, I think maximizing the value is really speaking to the tremendous efforts of a very involved team of data scientists, managers, and curators that are working on the data. But to that point, the idea behind RDCA-DAP is really to create that integrated database for all rare disease, getting data from all sources, registries, industry, clinical trials, and then converting all that data into the same format to maximize the value and the utility of the data. So every little piece of data that we get is really looked upon by the data scientists that we have, taking a deep dive in the data, extracting the maximum value, and then making that available to researchers. There's a key aspect about RDCA-DAP is the potential that the database is going to open more and more for lessons learned and cross-disease discoveries. We were just discussing a task force in rare mitochondrial disorders that we're trying to launch with my colleague Amanda. Mitochondrial disorders, we are starting to get data for Leigh syndrome thanks to our collaboration with the Cure Myeloid Foundation. And so we're hoping to address these disorders. But rare mitochondrial disorders have an ataxia component, a seizure component, some have a neurodevelopmental component. And that speaks a lot to me because that's what RDCA-DAP is about. We have ataxia data, we have rare epilepsy data, and we also have a lot of expertise in how we can analyze, how to track patients that suffer from ataxia, for example. And that's going to help us address other rare diseases. And so that's really what this platform is about and how it's going to help accelerate efforts. Well, at this moment, we are developing disease progression modeling for 2 rare neurodegenerative disorders, one for Friedreich ataxia and an effort for progressive supranuclear palsy. And I think as we go along and develop more efforts for dementias, for example, we're trying to work and launch an effort for FTD, we're going to have more and more opportunities. And I believe, Klaus, we had a discussion last year about some of the models that when we develop a model for Friedreich, we see some redundancy or some kind of redundancy already seeing that we can maybe adapt, if not reuse, adapt some of the modeling that we develop for one disease to facilitate the development of models for another disease. So I like that a lot. There's a lot of more complexity than just, oh, we did that for that disease, we're just going to turn that into the same thing. No, it's not that. But I think that speaks to what we're trying to achieve here in terms of facilitating future efforts and really advancing our progress as we speak to disease modelization and developing drug development tools.
Perfect. That's a key point that you raised about the notion of the cross-learnings from one condition or family of conditions to another. And that's a perfect segue to Soren, because you've been also hard at work figuring out how the learnings from the PKD effort can be applied to many other rare and orphan conditions that have kidney involvement. Can you comment on that a little bit?
Sure. Thanks, Klaus. Yeah, and thanks, Frank, for your continued support and leadership for PKDoc. Yeah, so we have leveraged what the PKD Outcomes Consortium has done over the years to accumulate data from registries, longitudinal studies, RCT type data, and we built a clinical trial simulator tool that we are taking through the regulatory fit for purpose endorsement process. But as it turns out, this particular tool, while having a few covariates that are specific to PKD, for example, the total kidney volume one, can be leveraged for modeling disease progression in terms of kidney function, for example, in terms of other rare kidney disorders, and the ones that we are looking at, as Klaus alluded to, are in the rare genetic kidney disorders like ADTKD, autosomal dominant tubular interstitial kidney disease, kidney disorders associated with diseases like Fabry, cystinosis, et cetera. And the bottom of this effort really lies our proven ability to work with stakeholders, to get data, to get people to sit at the same table, to talk about what are potential, endpoints and outcomes we're looking for, and to, model those based on the data that we receive. And I think, the ability to work with multiple stakeholders in this neutral environment, I think, cannot be overestimated. And yeah, that has been the power of building these collaborations and having expertise in building tools that we can translate to hopefully other indications outside of PKD. And we're very excited about that, and I'll talk about it more in our showcase presentation this afternoon.
Perfect. Thank you. Now, Kevin, you've seen two important sides of the FDA, right? So the Office of Clinical Pharmacology and now a clinical review group. Really what we're talking about is under the umbrella of a concept called model-informed drug development. And so can you comment on your personal experience at the agency with how the concept of MIDD has grown and matured, and maybe also talk a little bit about both the MIDD pilot and the Fit for Purpose initiative?
Sure. So when I started, I think the approaches that we're talking about, these quantitative approaches, were something that was seen as a specialty where you would bring in a few people to help you if you had a problem. And there was a small group when I joined that was working on these problems and these kind of models. And we were sort of a small specialized group. And that's changed over the course that I've been there, where it's seen now as something that is important in the planning stage as well. It's not something that you come in at the end and you try to make sense of data that has some issues. This is something that is integral to the whole process. And when you start to plan a trial or a program, you start thinking of how to analyze it, the trial once it's complete, and how you could apply these approaches to be more efficient in the trial design and collection of the data and the analysis as you go on. To your other points, the questions you had, so we have had some success with these approaches over time, and that has led us to the MIDD program. It was a pilot program in the last PDUFA. I think we're hoping it'll be a stable program now in the future. But that's an opportunity for companies to come in with a modeling approach that maybe is going to require more time and effort than a standard kind of type C meeting, and it might require more expertise from people who do more of the quantitative work. So what we do is we offer the opportunity for a sponsor to come in and have that opportunity to talk with the experts in the agency about their approaches to model-informed drug development. And as part of the program, they get a first meeting, we have a discussion about it, we provide our comments and feedback, and the sponsor has a second opportunity to come in and have a follow-up discussion on that. And I think the program has been going on for a few years now. It's successful. We've had actually had a few drugs that have, or indications that have been approved that have gone through that pathway. So we're open to that sort of back and forth with companies early on in the process. And the other approach is the fit for purpose. And I think that provides us a way to see the integration of the data and the models with a fit for purpose indication, if you will. So I think the standard approach that you would think of when we interact with sponsors is that it's possible that we see a certain sponsor, they have their own approach, their own data set, their own simulation or tool, and then we see a second sponsor that has their own approach. And for us, it's a challenge to consider those approaches in the span of 40 days and from different companies, and we're aware of some data that they might not be aware of. So the Fit for Purpose program and what you guys have done on some of your approaches has been really to get the people together early on to talk about the tools that can be helpful in a certain disease area. And I think one of the strengths of that program and some of the work that has been going on in some of the topics you've heard today is really to get people on the same page. And this isn't just a tool at the end where it's a button, you click it, and you get some output. It's really about the process. I think when you start to embark on this kind of project, you really have to be transparent about what are the assumptions we have to make, where are the areas where we don't have the data and it's important to get that data, or potentially where are there parts where the data that has been collected maybe isn't the optimal way of doing it. And I think as part of that, you learn the best way to do it in the future. And a second comment about the tools are that it's not a static tool. This is not something that you go forward and it's a tool, it gets fit for purpose or some kind of qualification. It's meant to be something that is changing over time. So as you get new trial data that come in, the tool or the approaches should change. So as we go forward, we hope that these tools can incorporate trials and successes and new standards of care and to evolve as they go forward.
Perfect. Thank you, Kevin. And so one thing that I want you to also elaborate on is the experience that you saw, even though that may not have been in a rare indication, but the effort that we run with an existing tool that had already been endorsed by the FDA as a clinical trial simulator. But during the pandemic, when trials were being run and the dropouts were potentially going to kill those trials, and how we incorporate some functionality to deal with and help sponsors inform how to deal with the dropouts during the pandemic?
Sure, yeah. So as you mentioned, there was a concern from the part of sponsors and by us that trials that were started before the pandemic and were ongoing, some of our trials, they last 2 years or so. So they could be impacted by patients who are missing opportunities to come to the clinic to get the drug or to have the endpoint. So it was important to understand what is the potential impact of that on trial power and the interpretation of the results. And we've seen sponsors have different approaches how to handle that data, whether or not to exclude something or to include all the data. And so I think what we did during the pandemic, or what you guys did, was come up with a tool to help understand how trial characteristics can change in response to patients who are missing trial visits, and what is the impact of that on the outcome, or how to interpret the data. So that has been useful for me as the trials have come to fruition and read out, and trying to understand the impact of potential missed visits or missed drug administrations in those trials.
Perfect. So Kevin wrapped it up with a key component, a key message, key take-home message, the learn and confirm paradigm. And it deals with the 4 pillars that I was showing in the beginning. You generate a piece of evidence, then you see how that evidence feeds back into the clinical context, how does that fit into the uncertainty, the regulatory pathway, and the evolution of drug development. So with that, I will open the floor for questions from the audience. Do we have any first takers so that we don't put Michelle to sleep? So while you get more excited about asking questions, regarding the learn and confirm paradigm, Alex, I'm going to pick on you once more. One of the things that is a key learning from RDCA-DAP is also the ability to diagnose specific areas for continued improvement in data collection. Not as an indictment about anybody having done anything wrong or that the data are low quality, but focusing on the opportunities and making those opportunities happen. Can you comment on that?
Yes. Once again, it's not me, it's the team, but it's really something that always comes in conversation, data standardization, and making sure you collect more and better data, and how we place ourselves in that conversation. And so each time we get a data set, it's a deep dive and a diagnosis on the data set that we receive, and so the team is able to feedback that to the people that contribute data to us and let them know where were the gaps in their data collection, where do we see areas where you can fix things, what recommendation we can make for your standards and improving your data collection. So we're very much in that loop, and I call that a loop because it's starting to arrive. We're starting to get contributors that share data to us early. The team incorporated the data, provided the feedback report. The contributor continued their collection, made adjustment, enhancement based on our recommendations. They also made improvements by themselves. And we're going to get that data. It's about to arrive, an updated data, and we're starting to see how we are really helping improve these data collections.
Perfect. So Ramona.
One thing.
Yes, please.
I would like to add to this that one of the limitations, and for those of you that just attended the risk showcase, I apologize because I'm repeating some of the things, but really going through the processes and features of the clinical trial simulation tool, there are some limitations to our knowledge. But going back to the rationale, like behind those limitations, is really all data-driven, the granularity of the data that we work with, because the model are fit on the data that we work with. So starting from those data, then we have more or less granularity on specific features, like the genetic granularity, the use of steroids versus not use of steroid, and the modality of use of steroid, and how much we can be precise and really include as much as information in those models really depends on how granular the information we start from is. And so I think filling those gaps is really a process that comes from, again, once more, broken record, but data sharing processes are really what's driving strongly these initiatives. And we're really grateful to those data sharing champions that have been contributing to the development of these projects.
And I'm going to pick on you. Yes, please.
I would just add another piece on the collections. For some rare disease, the data core elements, or what needs to be collected, can be ill-defined, and some communities have some collections, but the conversation is starting, and the idea is because we can aggregate your data, we can make the most of it and then help you define what you should include in your data collection. And part of that is also a plug to Ramona on efforts that are ongoing where we're going to be able to help potentially inform COA selection, and now you can use pre-existing COAs to collect data in rare diseases that don't have a specific COAs. I think we have some opportunities that are rising from that front too, if you want to.
Yeah, I was going to give myself a plug, because you kind of started to answer my question. But, you know, at CPAP, we've been very successful at reusing data. Is my mic on?
No, I don't think so.
All right, I'll just stand this way. At CPAP, we've been really successful at reusing data, but data are collected for a lot of different purposes. There are natural history studies, the data that are collected in a healthcare setting, data that are collected in clinical trials. Those purposes often, and, you know, specifically around clinical trial simulation tools, they're not collected usually to create clinical trial simulation tools, or if they are. So I just wanted to ask the panel, like, if you had a wish, is there something that's missing that people generally are not collecting that you would like to see collected that would help accelerate this field?
Frank?
I would just say there's a step missing, and that step for me seems to be at the very earliest stages, engaging with the patients and understanding what they're most interested in having treated, not their disease, not themselves, but what aspects need treatment and need to see an effect. And the patient foundations and others like NIH and other people that are creating these registries, that they engage with the regulators to say, well, if I wanted something to help this, what do I need to do? Is it a COA? Is it a questionnaire? Is it a biomarker? Is it something else that needs to be collected or integrated? And then do that at the very earliest stage. The other important ask, I would say, and this is why I'm sort of re-engaging with CPath in my new endeavors in Cushing's disease, which is another very rare disease, endocrine this time, which is actually what I am, an endocrinologist, is I've seen now 4 drugs approved since I first was introduced to the disease 30 years ago. And none of the programs have really followed the same path. And they're using different tools, and there's a lot of dissonance and you know, application of COAs that would not be approved other than the fact that it's an extremely rare disease and very difficult to study. How can we align the data that Cushing's patients are interested in with the data that patients with adrenal insufficiency or other disorders that are very closely related to develop tools that will apply more broadly, not just to endogenous Cushing's disease, which is caused by a tumor, but to the Cushing syndrome that we cause as physicians when we try to treat rheumatologic disorders or asthma or things like that. So I'm very interested in seeing how, at a very early stage, we begin to integrate these things into the registries or the databases and build the drug development tools that are going to be needed, not only to look for efficacy, but also to look for safety or toxicities of those agents.
I just want to say, I swear this was not planned, but this is exactly what I'm going to talk about. That we're going to talk about in our presentation.
Okay. I'm sorry, I will miss it.
That's why we set up the meeting the way we set it up. So, Frank, you mentioned a really important point, and that is going to bring me back to you, Ramona, because one of the things that was a prime example of that continued learning and the incorporation of the voice of the people with the actual experiences that matter, and how expanding the collaboration with colleagues like Sarah, for example, led to additional modeling efforts that are looking into other types of metrics, specifically imaging markers.
Yes, and this is a very exciting project that the DDISC is working on as well, in collaboration with the Emerging DMD Consortium and the collaborators at University of Florida. We are developing a clinical trial simulation tool based on imaging of fat fraction. So fat fraction infiltrates the muscle when the muscles start being wasted, and we know that there is a correlation between fat fraction and muscle waste. So basically, looking at the progression of fat fraction infiltration in the muscle can tell us how the disease is progressing, And having a prediction of the speed of this progression is really important. So for now, we are starting with quantitative solutions that tell us how the trajectory changes. And so we are building a clinical trial simulation tool based on the fat fraction trajectory, but what we want to look next is how much this trajectory correlates with the function. And that's something that will come next. But at this point, it is really important for us to really collect the data and the evidence that tell us that there is association between fat fraction and disease progression in Duchenne muscular dystrophy. The same has been done in other dystrophinopathies, for example, Baker muscular dystrophy. And also, fat fraction seems to be a very useful tool to also outline carriers in Duchenne muscular dystrophy because there is a component that is not really symptomologic in terms of loss of ambulation, but fatigue can also be associated with fat fraction deposition in those carriers that sometimes don't even know have the genetic mutation. So really using these quantitative tools to start with exploratory activity toward these very powerful instruments is what we're looking forward to accelerate next.
Yeah, perfect. And Kevin, I'll probably ask you to close us out with your reflection from your experience at the agency of how sometimes there's this depiction that the agency has a bunch of priorities, but they seem like they're separate and kind of on its own, like, you know, MIDD, PFDD, DHTs. And so, and especially with the notion of clinical meaningfulness and patient-focused drug development with MIDD and how the two are integrating, can you comment on that?
Well, I could say that for all of those that you just had mentioned, it's not, well, I could say, for example, for the MIDD program or Fit for Purpose, it's not that you have me and the modelers in a room who are determining is this fit for purpose. It's a team effort, statisticians and clinicians and everybody on the same page and together looking at these issues. This is never, you know, a silo where just a group, you know, DHT looks at this. It's always in the context of what is important for the clinical need and clinical trial design. So it's always the case that this is viewed in that perspective.
Perfect. All right. If there aren't any outgoing questions or comments from the audience, with this, I'm going to thank the panelists and close this session. So thank you, guys. And with that, I have the honor to introduce my friend and colleague, Lindsay Murray, who is the Executive Director of the Rare Disease COA Consortium. And she's going to give us a cool overview of a resource that is now ready for prime time. So thanks, Lindsay.
Tall, big step, short skirt. This is not the way I want to start this conversation. Okay, hopefully you all can hear me. Hopefully you can see me over the podium. I'm feeling a little short today. But, well, I'm Lindsay Murray. I'm the Executive Director of the Rare Disease Clinical Outcome Assessment Consortium at CPAth. And yes, I'm here today to roll out our Rare Disease Clinical Outcome Assessment resource, which has been 4 years in the making. So this is an exciting day for us all. All right, I've been told I push the button, and it will go to the next slide. Oh, yeah, that's me. Okay. There we go. All right, so I actually joined CPAth when CPAth got a grant from FDA to start the Rare Disease Clinical Outcome Assessment Consortium. And they brought me on to kind of figure out what that meant and how we were going to do that. But FDA was very firm with us, and some of you who are in the room will remember what they really wanted at the end of this day was what they called a common resource of COAs that could be used across multiple rare diseases. So we had to really stop for a minute and figure out what would that look like. There are 7,000 plus rare diseases. How do I create one common resource for all of those tools, or all of those diseases, rather? I'm pushing the big green button. Okay, there we go. So we spent about 2 years with FDA seed money kind of pondering those questions. How are we going to organize this? What are we going to build? How are we going to build it? Who should be involved? And in January of 2022, we actually launched as a separate individual independent consortium funded by member fees, which was an exciting milestone in and of itself. But really, the mission, again, was to create this pre-competitive, multi-stakeholder space where we could really think through the challenges in rare disease research and identify scientifically sound tools and methodologies for collecting the clinically meaningful outcomes data in rare disease trials. You know, just a minor, small task, small lift. So we currently have 21 member firms that include both a mix of biopharmaceutical companies, and then we have 2 patient advocacy groups, NORD and RareX Global Genes, and then we also are partnered with colleagues in Europe with the International Rare Disease Research Consortium. So that's been great to have all these different minds at the table together. And even better, we had industry co-director, my industry co-director, Dawn Phillips, is here. I don't know where she is, somewhere hiding. There she is, she's in the corner, who's got a great deal of expertise in rare disease clinical trial space, kind of helping us guide where we're going. We have an official FDA liaison who's been wonderful to work with. And then we had some really fantastic clinical experts that we were able to bring in to help guide our initial efforts, who were kind of, I called them my boots on the ground. They were in the trenches doing the actual measurements in clinical trials, as well as being able to bring representatives from a number of different centers from and NIH into the mix, as well as the Patient-Centered Outcomes Research Institute, and we've also had involvement from several folks from the CBER side of FDA. So we've been able to bring all of these really smart, fantastic people together in one place. So we started thinking, how are we going to build this resource? And where are the biggest challenges? And so very quickly, the consensus came to, we're struggling the most in kids. Pediatric, non-oncologic patient populations is where we are having the most trouble coming up with good measurement strategies. And that was from both FDA and regulatory bodies and our sponsor firms coming to us and saying, we don't know what to do here. Like, let's get together and think about this. And as I mentioned, you know, with 7,000 and growing rare diseases, it was quickly obvious we couldn't go through every rare disease and say, these are the 5 best COAs you should include in all of your trials. That was not realistic and really not a good use of anyone's time. So we identified pretty quickly, the way we were going to approach this was to use a domain approach to identify COAs that may have applicability across multiple rare diseases. And we decided to start with a couple of items that fell under daily functioning, because when we talked to families, when we talked to our sponsor firms, that's where they're really seeing, you know, the functional benefits of treatment. That's what families wanted to see, not just, you know, can my child tap their fingers so many times, but can my child do a button? Can my child hold their spoon level enough to feed themselves? Because as the mom of 3 small children, I can tell you it matters a lot to how my day goes once they hit those milestones. So we started there with self-care, gross motor function, fine motor function, and communication and language. And we really developed a strategy for how we were going to embark overall as a consortium. Obviously, the first goal was to establish and then expand this rare disease clinical outcome assessment resource, but really also wanted to focus on, we have all these experts in the room. We really want to promote collaboration and education and make sure we're learning from each other, because in the rare disease space, as it came up multiple times this morning, it's not any good if you do a clinical trial and it fails and all that information is lost. You know, we need to be sharing our learnings. We need to make sure that we're propping each other up, honestly. And so we also want to advance solutions for methodological and measurement challenges in rare disease by engaging all of these experts and making sure that we're disseminating that information, educating, and connecting with our partners in research. All right. So this is kind of the work structure of the COA, the Rare Disease COA Consortium. And as no surprise, in the center of this is really building out this resource. As I mentioned, we started with 4 subdomains of daily function. We're actually expanding now into pain and sleep disturbance and impact. And that's really the core of what we do. But again, we had all these experts in the room, so we really have tried to attack some of the methodologic challenges as well. You know, I think it's been mentioned several times, rare diseases are very heterogeneous. They have patients within patient differences, between patient differences are huge and really impact study design. So we did a literature review to really look at kind of measurement approaches that had been used to address that in clinical trial design. And we identified six methods, and then we actually linked those to FDA approvals by type, and then thought about it through the lens of how might these apply in rare disease research programs. And that publication actually came out in Value in Health earlier in this spring, and I have a link for it in the next slide. We started November of 2019, and then 2020 hit. This little thing called COVID kind of made life really difficult for all of us. And our sponsors came together and said, we are struggling. So we pulled a team together and looked across the lifespan of a clinical trial where COVID was impacting the disease, it was impacting the ability to conduct a trial, and then how people were mitigating those challenges. And we put together a really comprehensive table with all the mitigation strategies, both in clinical trial settings and in the clinic. That's all published online if you guys want to look at it still. Hopefully it's now not needed, but you know, it stuck around longer than I wanted it to. But it was a really amazing opportunity for us to show that we could be very nimble and flexible with and address the needs of the community very quickly. So that, you know, that was within 6 months of the consortium addressing that this was an issue. We had a webinar pulled together. We had some actual ideas for how to mitigate these challenges and move things forward. More recently, we started an endpoint creation for gene therapy team. We were really looking and thinking through if you're how you structure a trial in gene therapy is not quite the same as when you're just you have a drug that's going to make certain aspects of the disease better, but is not going to be, I hesitate to use the word curative, but it's not going to be disease modifying in the same ways. So we have a working group that's working on kind of best practices for that, and how, based on looking at how some of the approvals that have come out so far, what they've leveraged, and seeing if we can draw kind of some learnings from that. We also have a qualitative data collection team because, you know, as it was mentioned earlier today, certainly from the statistical side, rare disease programs are always underpowered. So, you know, qualitative data can add such rich experience data to a clinical trial setting when it's collected systematically, and really help move the needle when there are cases like Dr. Baracchio was talking about earlier today, when maybe the primary endpoint didn't do what you expected it to, but if you have qualitative data that helps kind of support, but patients didn't just, you know, walk a few extra feet, they actually felt more stable, they didn't feel like they fell as much. Those kinds of inputs can be difficult to quantify, but if you have rigorously collected that in a way during your clinical trial so that you can compare, say, if you've got it, placebo versus non-placebo, and see if those effects seem to play out in the qualitative data, that totality of evidence package is now way stronger than just, they moved an extra 3 meters. So we're working on actually 2 papers in that group to really help support why you should be doing qualitative research in rare disease trials, and then really more practical, like how to, because there are challenges doing qualitative data collection in rare disease trials. And then the last methodological issue here I wanted to just highlight briefly is, so NORD was in a subawardee on our original grant from FDA, and it was a fantastic opportunity for us to work with them and some of their I Am Rare registry partners. And so we did a brief survey and then followed up with some structured interviews to really get a sense from registry developers, why are you building these registries? What are you including? How are you making the decision on what to include, measure in terms of measurement? What are you measuring? How are you measuring it? And what are the pain points? Because I think it's very obvious that the patient advocacy groups and patients, when they're inputting that data, they want it to be used for something. They want it to be clinical trial ready. They'd love for it to stand as a natural history leg or, external control arm. But frequently the way the data is collected doesn't make that possible in reality. So seeing if there were places that potentially CPATH could help close that bridge. We might not be able to completely pave that road, but at least improve the quality of the data that's coming out of patient registries. Okay. So as I mentioned, this is the Value in Health publication that came out about the measurement approaches, and then we have the launch of the COA resource, which you're all here for live. Mark the day. All right, so I want to take a brief moment before I launch into showing you the resource to really just kind of take a step back and what is the resource? The Rare Disease COA resource is designed to provide information on published COAs that have the potential to be used to support efficacy endpoints in treatment trials for rare diseases, which sounds easy, but but what I want to kind of take a step back and really make sure everyone is grounded in is, so there's COAs encompass 4 types. Patient-reported outcome measures, I think, are the most commonly used, followed by, and then there's observer-reported outcomes, ClinROs, clinician-reported outcome measures, and performance outcome measures. And all of those COAs together are supposed to measure how an individual feels, functions, or survives in the setting of a clinical trial. I would say, by and large, most people immediately think of PROs when they think of COAs. 50% of rare diseases occur in pediatric populations. I promise, as the mom of 3 kids under 10, none of them can reliably report on anything, and they don't have a rare disease. When I see my daughter hit my son, and I said, "Did you just hit him?" And she says, "No." I don't know that we can include that in a clinical trial setting. Some reporter issues. So we're not looking at including PROs in pediatric populations. And I will emphasize there too, I mean, we also, I'm making light of it, but you have very young children as well who are pre-verbal that you wouldn't expect to be able to respond. So when you think of COAs in pediatric rare diseases, stop thinking about PROs. You're not going to use them in most cases. We are going to be talking about OBS-ROs, ClinROs, and performance outcomes measures because I can't ask the patient. And that's just the reality. The other thing I really want to make sure people are grounded in, this resource is not a library where, I'm going to go and select this tool, throw it in my program, and it's going to work perfectly. The FDA has issued 4 guidance documents on how that's not going to happen. And the truth is you have to understand your disease. You have to conceptualize the clinical benefit that you think your treatment is going to have on the disease. And then you have to really pick an outcome. What kind of outcome? Again, not a PRO. And so how does that, not being able to get self-report, how does that impact your ability to measure certain things like pain and sleep quality when you can't ask the patient? How are you going to do that? And then you have to decide, is there something existing or do I have to make something new? And you still have to find support and evidence to show that the tool you're looking to use makes sense in your patient population. I don't think any of us want to have a trial where, Executive functioning, yes. Median age of my population is 3. I promise executive functioning is not going to move in your trial. So you really have to think through and ground what you pick in your understanding of the disease and your understanding of what your treatment is supposed to do, which are enormously challenging in rare disease, and I don't mean to make light of that, but can sometimes get lost in the message. And I just really want to make sure that people keep this in mind when they're looking at tools that are included in the resource. So we have developed a pretty robust methodology for selecting COAs that go into the resource. We have a domain and prioritization and definition step, and then we conduct a landscape analysis for COAs for the selected domains that not just in clinical trials, what's being used in clinical care, natural history studies, across the board, to really make sure we're not missing anything. And then we kind of winnow that down based on some, honestly, common sense guidelines. Is this actually measuring what I think it's supposed to measure? And quick review quickly tells you no for a lot of things. And then we do an in-depth gap analysis where we really look at each of the tools against evidentiary standards that have been established through the 4 FDA guidance documents, through EMA guidance documents, and other regulatory bodies, and really put these tools under the gun and say, are they going to measure up? If I submit this to regulatory bodies, are they going to measure up? No tool has them at all, by the way. I have 15 years as a COA developer and never managed to hit every box. There's always something that doesn't quite fit. So that's not the expectation, and I don't think that's not, as a COA developer, I would never expect that. I don't think that's regulators' expectation, but you need to make sure that the tool you're using at least hits the minimum of it's measuring what I think it's measuring and it's doing it reliably. And then we have a consensus process. I've been blessed in my consortium to have a number of internal experts who have worked in pharma for a long time and have seen rare disease programs through to approvals. We have a number of folks who were physical therapists in their past lives and actually implemented many of the tools that are used in rare disease trials, as well as our clinical experts. And as well as our regulatory colleagues, honestly. And we've all looked through the data and kind of picked which tools we thought should be included in the resource. And then we convened advisory panels with external occupational therapists, physical therapists, and caregiver representatives to double check our work and make sure, did we miss anything? Are there other tools we should have considered that somehow got missed? And honestly, in that process, we also gained a lot of information from the caregivers around what they did and didn't like about some of these tools. When we were looking at the gross motor function tools, many of them include aspects of ambulation, and our caregiver representative said, my daughter stopped walking 4 years ago, and every time we participate in a trial for our disease, this questionnaire is included, and it's like pouring salt in the wound for me. She was really upset, and it really bothered her. And we talked about it as a group afterwards, and it's like, this is a really amazing opportunity for us to also highlight where all of us can do better to educate each other on why we're including certain tools in our trials. Sometimes we have to include these tools. The population goes from ambulatory to non-ambulatory, and we have to capture that inflection point. But as clinical trialists, we need to make sure we're engaging with our patient communities so they understand why they're being asked a questionnaire that asks about ambulation when their kid isn't ambulatory anymore, and make that case clearer so that they're more engaged with us. And it really just struck to me how much this really needs to be a collective effort for everybody. And so we've really tried to actually pull that contextual information into the resource and make notes of places where the caregiver said, I hate this tool. We didn't put it that way, but it's in cases where tools are included aspects like this, you might need to do some additional outreach and education to your patient advocacy partners to really make sure that they're engaged the way you want them to be. And then we've made all of this information available on this website that we've launched, and you'll have access to it all. So the tools that have been included in the resource represent tools, I'll put a few caveats on this. So they represent tools that we could find. They had to be in the published literature. I know there are tools that are being developed. Many of you are probably developing them in your individual programs, or I know there are a number of patient foundations that are creating their own COAs. But if they haven't published anything on it, we couldn't find it. But this will be an iterative process. I was speaking with somebody last week, and I already know one tool that's being developed out of Duke, the Observer Reported Communication Assessment, which was originally developed for Angelman, and now has one of the core outcome grants from FDA to expand into 12 other rare diseases. Hadn't published anything, but they came and presented to the consortium so that we were aware of the tool, but it's not included in the resource yet because the publications hadn't come out. So you will see tools that we could find. And there are also cases where there's historical COAs that have been included where they don't meet many of the evidentiary standards. They've been used in the communities for so long that you have to include them. And yes, there probably needs to be some modification and some adjustments to them, but there's so much evidence based on them that they help kind of guide where we're going, and in many cases also provide the ability for us to look at them, the data against normative standards, so how your kids in your population are doing against what norms would be to help interpret your trial results. I want to emphasize, the COA resource is not a core outcome set. So a core outcome set would be you go to self-care and I give you the 3 COAs that you're going to use always in to measure self-care. That is not what this is. You guys are still going to have to really look at the tools that we've included and think back to that framework and select the tools that best fit your programs. But what we have done with this resource is I like to call it the best ofs. We've collected the best ofs. We've looked at everything that's out there and really put them through the ringer of evidentiary standards and selected the tools that are most likely to work in clinical trials for rare diseases. And then it's your job as the user of this resource to go through and look at the 10 self-care items or COAs we've identified and decide which one makes the most sense in your community. Which one is measuring things like being able to do a button or pull a zipper, and does that make sense in your community? And is that something your treatment is going to target and be able to see effect on? You're still going to have to go through that process. But now you don't have to look at the 600 COAs that for self-care and go through all the steps to decide which ones might work in your treatment trials and your population. So the resource can reduce the time and cost it goes into deciding which COAs to include in your rare disease trials. As someone who worked for a CRO for a long time, I can tell you I made a lot of money doing these lit searches for folks. So we've done that for you for these domains that are available today. And so we really want to be able to help you select the appropriate COA for your individual research programs. And then the other thing I'd really like for this resource to do, and we'll see how this plays out, but I'm really hoping that patient advocacy groups will be able to see the COAs that we've included. Some of them are observer reported and could be easily administered in a patient registry. Some of them are not. But at least have an idea of how to best measure some of these domains in a better way that's more robust, that gives us better data out of registries. So that's maybe a longer term goal for me for the resource, but that is something I'm really hopeful about. Again, as I mentioned, right now the first iteration of the resource includes gross motor function, fine motor function, self-care, and communication and language. And then we are expanding. We're looking at pain interference and pain severity, sleep disturbance and sleep impact, and we'll be expanding over time. So this is what it looks like if you go to the Rare Disease COA resource. For right now, Google is a little behind, in my opinion, but it's not searchable in Google. Apparently that takes a few days. But if you put in the full website, you can access the resource. And you'll see we have a number of tabs there, which COAs are in the resource, frequently asked questions, links to information, all of that is there for you. Again, you will need to put the whole, the whole web address there into your browser to get it at this point, until Google catches up. And I will say as well, just this is not optimized for doing on your phone. So if you're pulling this up now on your phone, it's gonna look funny. There's a lot of information, and when later on this afternoon, we're gonna be doing kind of a live walkthrough of it, and you'll see, if you're looking at tables next to each other, I think we can all agree 3 inches wide, you're not gonna get a lot. So we really focused for this first initial round on optimizing it for desktop. So again, you'll be able to go and do COA searches. You can search by name. If you have a specific COA that you want to know about, you can pull that up. If there are different versions of a COA, many of you are familiar with the Vineland. If you put Vineland in, you will get 5 different versions of the Vineland that you can then compare to see what are different between the 5 versions and what you might want to work with. If you don't know specifically what you want and just kind of want to take a peek around and see what COAs have been included up to this point, if you click on COAs in the resource, there's a table with all of the COAs that have been included thus far with links. If you click on the links, it'll give you all the information from the gap analysis, which is honestly a lot of information. And all of it is available for free. So that's a big bonus. There are other databases out there that will provide some of this information, but you pay a lot of money to get access to them. So that's something that we are very happy to be able to offer. So as I mentioned, we have a showcase later this afternoon where instead of slides, I'm going to have a computer and I will just walk people through how you interact with the resource, and we'll have a chance to actually kind of get into the nitty-gritty of the resource for those of you who are interested. That's tonight, or this afternoon, 4:15 to 5 in the ballroom, E75. So I think that's actually the room next door. Okay, thank you, Laura. And then before I end, I know you can't see all of the different consortium. These are all of the rare disease efforts we have at CPAth, and I know Colin has emphasized this earlier today as well, but none of us are operating in a vacuum here. CPAth has a tremendous rare disease program, and I'm thrilled to be a part of it, and I feel like sometimes it's easy to think, oh, okay, they're doing that, those folks are doing this, but the truth is, we're all talking to each other in the background. I'm having conversations constantly with our quant med teams about data analyses that they're working on. I'm talking to D-Risk about potential COAs to include in the Duchenne world. I'm talking to CPTA about outcomes in those, that can be used in those trials. There's a lot of cross-consortium and initiative collaborations at CPAth that we really, this is a relatively new that we have so many, but I'm really excited about our opportunities here to really leverage information and having all of these people together at the same time in the same place, and kind of getting to the point where Colin had in one of his slides where we have all the right people and at the right place at the right time to really make some big improvements in rare disease research. So I'm really delighted about that, and I hope this resource helps get us that much further. So with that, we have a few minutes for questions. Do you have a question? Okay.
Hi there, Jessica Bohanowicz from the Foundation for Prader-Willi Research. We are one of those organizations who have a lot of very specialized caregiver-reported outcomes, but a lot of them, although they are used in the pediatric population, they are also extended into adults as well. So I was trying to understand a little bit about, I understand that you're focusing on pediatrics right now, but are the surveys, is there like an age cap limit on the surveys that you're generally looking at, or are you looking at ones that just pediatric, but also span pediatric into young adult and adult?
So our searches generally went from, I think we had it go from 2 to 18, were kind of the general search parameters. Individual tools do sometimes go beyond that, and that wasn't an exclusion criteria for, yeah.
Okay, thank you.
Oh, Ramona's got a question.
I have lots of questions, but I'll just ask one, simple one. You were talking about the inclusion of COAs that were older, that are sort of a standard care, that you had to include them. Do you have a note on your resource that says, this is an older one that we're including for standard of care? Because a naive person coming in might think, oh, look, it's in the resource. It must be one of the really great ones.
Yes, yes, there are. We do have like a comments, but yes. Short answer, yes. In a way that's as nice as possible. I didn't want to, I don't want developers coming after me with pitchforks. But there are tools that are being used that, really are there just because. I think.
Hi, Susie McCune from PPD. This is a fabulous tool and a great resource. I know that you've broken it down by, let's just take motor, but clearly infant motor, different. I haven't had a chance to get into the specific tool. Do you have that information, the specific tool, in terms of the ages that are validated for those? Okay.
Yes, and that's, again, why this is not like a click-and-go library, because because of those issues, we have tools for motor skills that go from when you're evaluating a 2-year-old to when you're evaluating an 18-year-old, and those look very, very different.
Absolutely, and even down to birth. So
oh, yeah, absolutely.
Yeah, no, thank you very much. It was a great tool.
Thank you for your talk. I am curious about, because it's all based on lit review. I've worked with RDCA-RND, who is trying to line up all of the outcome measures that they've used, and what's often not in lit review is the negative data. So those COAs that have been used that they haven't measured effectively and have been abandoned, and we don't know about that. And so sort of for the future, learning from KOLs and how we can maybe implement that into the resource, those.
Let me see if I was having a hard time hearing you, but so COAs that didn't get included but might have worked, or COAs that don't work?
COAs that don't work for particular, say, nonverbal children, right? So we, as a good example, right? Communication is hard to collect, and I've seen in RDCA-RND several examples of where during the U54 natural history study that some disorders just stopped collecting Vineland because there was no measurable change being seen in certain disorders. You may not see that reflected in the literature because it was never published because they stopped collecting it.
Right, so that's where the consensus process really became crucial, was we had the experts who have used these tools, who brought that experience and said, okay, Vineland's great unless you have a population that stops being verbal. And so that's a note on Vineland, like, there's a caveat here. It's a great tool, but not if you're not verbal. And then you might want, so we included also tools that included aspects of nonverbal communication. Again, getting back to that, you're still gonna have to select the tool that's right for your patient population. There is no magic bullet, unfortunately, but.
Thank you for the great presentation. I have 2 questions. So you mentioned recently developed COA may not be included in the data source. So I was wondering how often the resource is going to be updated?
That's to be determined. I have a 2-man team, a 2-woman team, actually, to be clear. No, but we're trying to stay on top of that. And one of the tabs that we have, I don't think we released it in this iteration, is up-and-coming COAs that we are aware of that maybe haven't met that threshold, but that people should maybe want to be aware of.
Another question is, do you recommend using this data source with other like COA library or databases like eProvide?
I mean, sure, you absolutely can. The information is going to be very complementary.
Perfect.
Yeah.
Thank you.
Uh-oh, now I'm in trouble.
Well, I was just going to echo that once you select something, you should come and talk to the agency about what you've selected. I mean, I wouldn't be me if I don't say that, right? If you haven't been around me at enough conferences. Because really, this is to help folks get that jump start, right? This is a very intimidating field, a rare disease. How do I pick the right thing? And where do I start, right? So really, this is meant to help get a jump started with some basic information that has been driven by the consensus process. Could you maybe, Lindsay, talk a little bit more about that consensus process to the folks? Because I do think that was an integral part of those discussions that were said, because the point that was made, we would actually say, that may not be good. And there was a lot of good expertise on those conversations. So could you maybe delve a little bit more into what really, this wasn't like a one time we met for a day. Like, this was a very lengthy process. So I was hoping you could maybe talk a little bit more about the process and the different types of disciplines that were involved.
Absolutely, yes. That's a great point, Michelle. Yes, so that consensus process really, it was a process. We had a resource development subcommittee that we formed, and we had our regulators at the table with us. We had, I'm gonna out Dawn. My industry co-director was also a physical therapist in a past life. We had folks who have gotten drugs approved in the rare pediatric population space. We had external clinical experts that we had brought in, so 2 neuropsychologists and a speech-language pathologist, and then COA development experts who all went through the gap analysis reports. And there were, I think for communication alone, we had 68 different COAs that we were looking at. So we went through every single one and looked at all of the information and we batted it around. The speech-language pathologist would say, oh, I use this all the time. And Michelle or Naomi would occasionally be like, yeah, let's not include that. It's not, we've seen that, it's not working. It's great for clinical care, not so great in the clinical trial space. Or sponsors would say, yeah, used that, burned bad, don't do that. And we really had that open dialogue with each other. It was, it was 10 or 12 of folks that are participating in my consortium who really sat around for months, honestly, going through all of these reports and then really thinking through, like, how were they being used and how successful had that been outside of, clinical practice? How were those being used in clinical care or in clinical trial spaces? And then we checked that with external advisory panels to make sure that we were on target, and pulled in additional physical therapists, occupational therapists, speech-language pathologists, caregiver representatives to make sure that we hadn't missed something, that a tool that they thought worked really well had somehow missed the screening process, and really ended up pretty happy with the results. But it was an iterative process. I think each of the advisory panels met at least 3 times. The resource development committee met every 2 weeks for, 6 or 9 months to go through the 4 domains. So it was a lengthy process with a lot of back and forth discussion.
Hi, I'm Karen Kaiser. I'm from Northwestern University. Thanks for your talk, Lindsay. It was really great. I had a question about condition-specific measures versus more general measures. So with this domain approach, were you prioritizing more general measures over condition-specific measures, and how did you handle that, given that, especially since some condition-specific measures may have less evidence supporting their validity and other things?
So I will say, I think we ended up with more generic measures overall, because there's more reported on them, but we certainly didn't exclude if there was a condition-specific tool that we looked at it and said, hey, this worked really well in this disease population, maybe it could be applied to something else, then it got included. Now, you're all coming in. All right, Dawn. I did call you out.
I just want to add that this isn't going to be like the thing that's going to answer all of your questions. You still have to go back to the roadmap and say, what are my concepts of interest and how do they map against this measure? And so it's meant just to give you a starting place to say, oh, I never thought of this one, because our discussions were so robust. And for these 2 people, maybe they had an incredibly positive experience with this very specific disease and this very specific age. And then somebody else had a very different experience and said, well, that had a ceiling effect for me, or I couldn't use it in this way. And so it doesn't take away your homework. It just gives you options.
Thank you, Dawn. All right, any other questions? Oh, they're all coming after me.
I couldn't pass up the opportunity. I'm Naomi Noble. I'm the Associate Director of the Division of Clinical Outcome Assessment at FDA and your official FDA liaison for the Rare Disease COA Consortium. I would gently suggest that Michelle Campbell is our unofficial other FDA liaison. You didn't need any more hats, Michelle, it's true. I just wanted to express to you and to the group here as well, just what an extraordinary achievement this is to finally launch this resource. And as I think Dawn so nicely pointed out, as Michelle did as well, it's a starting point. It's a starting point for a conversation, but the efficiencies that you've created here for this resource to make it no cost to everyone who wants to access it, I think is just an enormous contribution to the whole of the rare disease space. And so huge kudos to you, and Barb, huge kudos to you as well for so much of the behind the scenes work in the day-to-day, which is not always terribly rewarding, but really important all the same.
Yes.
And to just, as well, the broader work that we're doing. So many of our gene therapy Endpoint group members are here too, and with our qualitative working group too, I think the whole of the consortium is really working to elevate, ideally, the knowledge base about how to use and implement COAs in the rare disease space, so well done.
Thank you. And for those of you who don't know Barb, say hi to Barb. She's my senior project manager for my consortium. She's actually the one who's honestly single-handedly responsible for getting the website developed and working with our sometimes ornery web developers. So she deserves a huge round of applause. All right. All right, Laura.
Okay, another great thank you to Lindsay for that informative presentation, and a big thanks to the audience, some really, really great questions. So from here, I would like to welcome Sergey Rakilin to the stage. He is our Executive Director for our Critical Path for Sickle Cell Disease. So he's going to share a little bit with you on endpoint selection.
And you do. Okay. Hello, everyone. Can you hear me? It's a great honor to speak in front of such a wonderful audience. Thank you for having me here. The title of the presentation that I have today is Endpoint Selection for a Disease with Highly Heterogeneous Manifestations. And I'm an executive director of the Sickle Cell Disease Consortium. Our consortium is relatively young. We're only 3 years old, so we're still trying to catch up with other more advanced consortia in developing models. So I will briefly introduce sickle cell disease to you and outline potential solutions in identifying endpoints for that particular disease. So sickle cell disease affects approximately 100,000 Americans and more than 20 million people abroad, worldwide. And there are 4 therapeutic drugs that are currently on the market that partially alleviate the symptoms of the disease, making it a clear unmet medical need. As of 2023, there are 33 pharmaceutical companies that are pursuing and developing treatments for sickle cell disease, and the number of companies developing small molecule drugs versus genetic therapy approaches approximately split in half. Half of the companies are developing gene therapy, making it a forefront of the development of those treatments that would be applicable for other rare diseases. So the points of today's presentation is to kind of give sickle cell disease presented in a form of the example of the identifying endpoints and identifying treatments that will be applicable to other rare diseases as well. What's important on that particular slide is that there are 2 BLAs that are currently being reviewed by the FDA, and we hope that sickle cell disease will be one of the first diseases that could be cured by the gene therapy approach. Now, I will show you the point of the next slide is a simplified version of the molecular mechanism of the sickle cell disease. What you can see on the left upper part of the slide is that a point mutation in the hemoglobin gene causes, rather decreases its ability to bind oxygen. As a result of that, hemoglobin polymerizes in lengthy fibrils, which become so long that it stretches the generally round red blood cells into sickle cells. And at some point, it causes hemolysis. Those cells break apart. This is a very important process because the half-life of the sickle cells is approximately 10 days compared to 40 days of the red blood cells in general, normal red blood cells. Now, as a result of that hemolysis, the occlusion of blood vessels occur. And that occlusion could happen anywhere in the body. It could happen in your hand, it could happen in your leg, it could happen in your brain. And that occlusion correlates with a great deal of pain that patients experience. Now, in the next slide, summarizes heterogeneous manifestations of the sickle cell disease that affect different organs. And you can think of that virtually every organ related to the penetrated blood vessels is affected. You can think of nervous system, and manifestation would be pain or stroke. You can think of cardiovascular system, where the acute chest syndrome will be prevalent. You can think of the reticular endothelial system, where anemia or hemolysis could occur. Musculoskeletal system could be equally affected in a form of leg ulceration. You can think of urogenital, gastrointestinal system, many other systems within human body that are affected by that disease. So this is a perfect example of the heterogeneous disease with heterogeneous manifestations that affect virtually every organ of the human body. Now get yourself in the shoes of the chief science officer or chief clinical medical officer of the pharmaceutical company. Try to come up with the clinical endpoints in order to develop a new therapeutical treatment for that particular disease. There are several options there. I divided those options into 2 groups. The first group relates to vaso-occlusive crisis, and they describe different aspects of that part. And the second group, I will talk about that a bit later, relate to surrogate endpoints or functional endpoints that are not directly related to vaso-occlusive crisis, but they're directly related to the mechanism of action of the drug itself. Let's start with the vaso-occlusive crisis-related endpoints. The first one, the most obvious one that we mentioned in the previous slide, is pain. The problem with the pain is that A, it's highly subjective. B, there was an observational study, what is called PICES, that was completed in 2008 and was completed by a distinguished professor, Wallace Smith, who specifically looked at the distribution of the pain within those patients where it occurred. And he, in this particular diagram that he summarized as a result of his study, you can see that the pain related to the vaso-occlusive crisis is only contributes only about 2.5% out of the 100% of the pain episodes ever occurred. What's important to that slide, and those 2.5% relate to the medical utilization. Majority of the paid events occur outside of the hospital and are being either self-treated or not recorded at all. It's also important to mention, looking at that particular diagram, that 45% of the patients having sickle cell disease have not, in that particular study, experienced any pain whatsoever. So what happened as a result of that is that pain, it's clearly is a very extremely subjective parameter, difficult to measure. The second problem is that there is a great deal of difficulty to distinguish chronic pain that results from the accumulation of end organ damage during the progression of a sickle cell disease from the acute pain that can actually be affected by a particular drug treatment. So you are struggling with a relatively narrow window, treatment window, that your drug has to resolve. Now, the first exercise that you can do is you can look at the, let's say, 5 recent drugs that underwent clinical trial, phase 3 clinical trial within the past 10 years. And the interesting pattern that failed and that succeeded. And the interesting commonality between those drugs that did fail, that are listed in that particular slide, is that many of them used change in pain as a primary endpoint. Look at the, those 4 drugs are listed on that particular slide with a reference to the clinicaltrial.gov. And you can, I highlighted with yellow definition of the measurement of pain. It's different, but the commonality between those 4 drugs, they all failed. Another potential approach to develop meaningful clinical endpoint for such disease with such a huge heterogeneity, both in the manifestations and the genotype and the phenotype as well, could be medical utilization. And that certainly was a mainstream for the drugs that did succeed within the past 10 years. Among those is hydroxyurea, L-glutamine, crizanlizumab. Now, the interesting part here is that, is the medical utilization ideal endpoint? Certainly not. And I'll explain that in that particular slide. So imagine that you're trying to, based on this slide that I showed you a few minutes ago, majority of the vaso-occlusive events occur outside of the medical utilization, outside of the hospital. So it would be nice to have a digital health device that would enable you to monitor changes in activity, for example, like it is shown in that particular slide. And what is shown in that slide is the difference of the activity in the activity of the patients without vaso-occlusive crisis and vaso-occlusive crisis. You can see that on the left side of the slide with the red arrow showing the difference. It's important to, and the data came from the Ellipsis trial. It was the observational trial that was run by Pfizer approximately 4 years ago. You can see the difference indicated by red arrow is incredibly narrow. And it only correlates to the difference, at the end of the day, probably within 4 hours before the participants of the study went to sleep. Now, if you look at the right side of the slide, where which is indicated by the yellow arrow, you can see that the difference is much more profound between the medical utilization, where the vaso-occlusive crisis occurred, versus the vaso-occlusive crisis that people experience at home or outside of the medical facility. There could be multiple reasons to explain why hospitalization generates such a profound difference. One explanation that you can come up with is that whether or not you're in pain or not, you wake up in the morning, you have to catch up the bus in order to get to your work. And the actimeter will measure the amount of that movement that you have to endure whether or not you're experiencing a great deal of pain or not. So the difference will be minimal. Whereas if you're in the medical facility, you're being monitored, you don't have to rush to work, you don't have to catch the bus. Maybe that explains the difference that you can see much more profoundly there. Nonetheless, the point that I'm trying to make of that slide is that however imperfect the medical legislation could be, whether or not it correlates with the changes in activity or changes in pain in the, that patients experience, the primary endpoint using medical legislation certainly works much better. But it's important that the third column from the left is the definition of the vaso-occlusive crisis that is used in those trials that were successful, that all include either the visit to emergency or medical facility or a visit to a medical facility in the second trial. So in other words, if you use visit to a medical facility, you can succeed with your drug however far or however relevant the action of the drug, the mechanism of action of your drug is to the medical utilization itself. All right, so if pain and medicalization are both imperfect as endpoint candidates, what could be a better one? Let's look at the functional ones, the ones that are not relevant to the vaso-occlusive events. And the first one that is on the list there is change in hemoglobin concentration. And that was the endpoint that was specifically used for the drug that was championed by the company Global Blood Therapeutics in 2018, and the drug name is voxelotor. The idea of the drug is that upon binding to hemoglobin, it restores activity, affinity of the hemoglobin to bind oxygen. As a result of that, there is no polymerization of the hemoglobin occurs, and then the shape of the red blood cells remain the same, and therefore the concentration of the hemoglobin goes up because the hemolysis rates are down. So then the primary outcome was formulated as a number of patients with increase in the hemoglobin concentration beyond 1 gram per deciliter, which is perfect. This becomes a surrogate endpoint that is clinically meaningful and that directly relates to the mechanism of action of the drug. The only problem is that other drugs do not, or sometimes they do not affect the concentration of the hemoglobin itself, so it's not universal. Could there be a universal approach that would be equally efficacious as the hemoglobin concentration? One particular biomarker that has been explored during the past 25 years quite extensively is a hemoglobin F. And hemoglobin F is a modification of the hemoglobin that is being highly expressed in fetus, and the level of expression of hemoglobin F goes down between 6 and 24 months after the baby is born. The difference between hemoglobin F and regular hemoglobin is the affinity to the oxygen of the hemoglobin F is considerably higher, and that could be easily explained. So when the baby is in the womb, it needs all the hemoglobin, all the oxygen that is in short supply. So about 25 years ago, the researcher Martin Steinberg developed the idea that overexpression of the hemoglobin F at the level of approximately 30% could alleviate any manifestation of vaso-occlusive crisis whatsoever. And based on that notion, based on the research that he did, many pharmaceutical companies started developing the approach where they decided not to fix the mutation itself, but instead they decided to overexpress the hemoglobin F to the level that would alleviate or could alleviate all vaso-occlusive events. And that was an approach that companies like Bluebird Bio took. One of the questions that we were facing while interacting with our stakeholders was, what is the threshold? What is the concentration of hemoglobin F that would be could identify ideal patients to be treated with gene therapy? So if you look at the diagram shown on the right side of the slide, you could think that hemoglobin F expression close to 0 will probably do not affect the frequency of vaso-occlusive events. As you move to the right, let's say hemoglobin F expressed in concentration 1% or 5% or 10%, would those patients be suitable for gene therapy treatment? And this is not a simple question, because many of the people have diminished expression of hemoglobin F close to 0, but there are a few expression level of hemoglobin F resides at fairly high concentrations. So for example, some have 10, some have 20, some have 30 and above. You could think of Sherpas who are moving, helping people to get high into the mountains. The expression of hemoglobin F in those people are particularly high. That helps them to cope with the dangerous environment they work in. And that is partially genetic. That could be partially be affected by the level of expression of hemoglobin F, and it varies from different people. So when you're selecting your patients for gene therapy trial, you better be sure what the level of expression of the hemoglobin F is in order to be able to work with the substantial treatment window and be successful with your drug. So when you try to answer that question, and if you take relatively large population of patients, which in our particular case was a cooperative study, which had approximately 3,000 patients with a sickle cell disease, and you try to run the correlation between the high hemoglobin F concentration and the frequency of vaso-occlusive crisis, you won't see much of the correlation there whatsoever. You start thinking, what's happening? Why is it not there? Was recently when a group from France came up with the conclusion that it's not just the concentration of the hemoglobin F per se, it is also the number of cells that express hemoglobin F at high enough level. In other words, what they used as a biomarker is the ratio between the concentration of hemoglobin F and percentage of cells, red blood cells, expressing hemoglobin in particular. And the way they did it, which is explained in that particular graph, which I'm not going to walk you through entire graph, but I'll explain it in simple terms. They did stain red blood cells with the antibodies against hemoglobin F. They calculated the number of cells expressing hemoglobin F, and then they also calculated the total concentration of hemoglobin once you lyse all those cells. The ratio between the two gives you a much better regression, what you can see on the upper left side of the, that particular graph when you compare the correlation between the percentage of the red blood cells versus the hemoglobin F concentration. And that what they did after that, they actually treated the cells extracted from 18 patients with different concentrations of hydroxyurea, and they tried to see whether or not the angle of that regression changes. And they identified relatively narrow window within which that change occurs. The reason I brought you this example is to demonstrate that even when you identify the readout endpoint like that, you can clearly see that that change is not directly proportional. There are multiple mechanisms involved in up and down regulation of the hemoglobin F that may or may not directly correlate with the frequency of the vaso-occlusive events. What do you do then? As a leader of the pharmaceutical company, And I started my presentation saying that there are 2 BLAs are being reviewed by the FDA right now. Both companies, that is Bluebird Bio and Vertex, both coming up, companies that developed the gene therapy treatment, came up with the same solution. They decided to use the probability of the affecting vaso-occlusive events. If you look at that, the upper part is the fragment of the primary, the definition of the outcome, primary outcome endpoint from Bluebird, and the bottom one is the similar one from Vertex. You can see the definition is almost the same. The proportion of subjects who have been affected by the frequency of the vaso-occlusive events. I'm just reading the Vertex one, the one on the bottom. And if you looking at the upper part, the one from Bluebird, is the proportion of subjects affected, complete reduction of the vaso-occlusive events between 6 and 18 months after the treatment. Keep in mind that both of these treatments are very much similar. Those are gene therapy trials, treatments that have been submitted to FDA right now. So as a summary, the ideal endpoint should measure how patient feels, functions, and survives, and should be meaningful to clinician for having value, be linked to the mechanism of action of the drug. None of the endpoints that are currently available work well for the sickle cell disease due to the heterogeneity of the manifestation and heterogeneity of the disease itself. Changes in frequency of vaso-occlusive crisis are currently used as a primary endpoint of the clinical trial, and the goal of the Sickle Cell Disease Consortium as part of the Critical Path Institute is to identify novel clinical endpoints and biomarkers that could contribute to the improvement of the success in clinical trials in general and in clinical trial design in particular. Thank you for your attention. Any questions? If not, I'll wait for those questions to arrive later. I was hoping that it could be some parallels between the sickle cell disease and other rare diseases that could be uncovered as a result of that presentation, but maybe it'll take time to uncover those parallels and define those questions better. Thank you.
Right, thank you. Okay, so we are at the afternoon networking break, so we have actually a few extra minutes for our networking break, so please take some time to network, mingle, charge your devices, check your email, do whatever you need. We'll meet back in this room at 3:45. There will be some snacks. They may still be setting up because we're a few minutes early, but there are some snacks out there. Be back here at 3:45, and then we'll round up the day with one more presentation, a couple more showcases, and some closing remarks and the networking event. So thank you. Okay, thank you, everyone. At this time, we would like to welcome Terina Martinez to the stage. She is our Executive Director for Critical Path for Rare Neurodegenerative Diseases, and I have a really hard time saying that, so I apologize. Executive Director for CP-RND, so welcome her. Thank you.
Thanks, and also just to add a little bit more difficult alliteration and alphabet soup to the mix, as my role at CPATH is the Executive Director for the Critical Path for Rare Neurodegenerative Diseases, Colin earlier walked you all through sort of the kind of like diagonal merging of HD Risk, our Huntington's consortium, and CPTA, our inherited ataxia spinal cerebellar ataxia consortium, into CP-RND. So if I gave you my entire title, it would include Executive Director, Huntington's Disease Regulatory Science Consortium, Executive Director, Critical Path for Therapeutics to the Ataxias. So please bear with me. What I will do is focus this particular talk on staging, disease staging, and the importance of biology and disease staging using Huntington's disease as a case study. But there are some really important conserved takeaways, cross-pollination, and opportunities to apply what has been implemented in the Huntington space to other rare diseases. And that's really the intent and focus of what I'll be talking about in the next few minutes. So the fundamental rationale for a staging system comes back to a lot of the themes that we talked about earlier today. We've talked about the industry perspective for drug development in rare disease. We talked about the extreme importance of clinical meaningfulness, measuring through endpoints and outcome measures, and a lot of the work that Lindsay was talking about with the resource for rare disease COAs, that you really want to assess how an individual feels, functions, survives. But we do also completely understand that that's only part of the picture, because disease is not just the sum of the parts of the clinical findings. It is really a complex milieu of the underlying pathophysiology, and you really need to understand the biology to get at that complex pathophysiology. So that's the underlying rationale and principle behind the disease staging system, to develop a framework to better understand the disease pathology, and then use that framework to standardize outcomes and better support interventional clinical trials. So this requires a data-driven approach to understand what the pathophysiological and clinical cascades are, where they overlap, where they are less overlapping. And so it requires a data-driven approach to develop the consensus methodology and then the algorithms to define the evidence-based staging system. We do have some precedent that we can look at for that, really focusing on the collective contribution of the biological, clinical, and functional assessments, and a few areas that can certainly be looked at as a playbook. We can start first with the early Alzheimer's disease. So back in 2018, the FDA issued their revised guidance for early Alzheimer's disease, and then the year after, the National Institute of Aging and Alzheimer's Association, NIAAA, published their framework to better understand the biology of early Alzheimer's disease. And from that, they developed what's called the ATN staging system. So this focuses on amyloid, tau, and neuronal injury, and the different components of that across the continuum of early Alzheimer's disease. With Huntington's disease, and what we'll spend another few minutes talking about here, is a framework called the Huntington's Disease Integrated Staging System, which again assesses within the paradigms of Huntington's disease the biological, clinical, and functional assessments. And then there's a very new staging system that is being developed. It's the neuronal synuclein disease staging system. This was a collaborative effort by several patient advocacy groups, nonprofit research foundations, such as the Michael J. Fox Foundation for Parkinson's Research and the Critical Path for Parkinson's, to develop a staging system for neuronal synuclein disease to help provide the biological understanding in Parkinson's disease and other related disorders. So there's an opportunity to look at the different lessons from these, but what we'll dive into in more detail now is the approach that was taken for the Huntington's disease integrated staging system. And really what I want to emphasize is that the staging system, when applied broadly across different diseases and specifically to Huntington's, should reflect the biological and clinical stages that can then serve as the foundation for stage-appropriate assessments and inform and optimize how to bring bona fide disease-targeted interventions. So just a brief overview for Huntington's disease so you can understand the relevance of the different staging components. It is a progressive, rare neurodegenerative disease with autosomal dominant inheritance. We'll get into a little bit of the genetics as we discuss stage 1, but it is highly penetrant, and this means that families that have Huntington's disease within the family tree have a risk of 50% inheritance of children getting Huntington's disease if a parent has manifest Huntington's disease. But it's very complicated. There's a paradox within neurodegenerative disease broadly that clinical diagnosis is required because until you present with movement disorders or with cognitive deficits, you don't know that you have the underlying biology already occurring. So we have to really back up. And one of the challenges with Huntington's disease is that it is slow progressing. And the transitions from pre-manifest, and I'm going to point out later, pre-manifest is a dirty word. We are going to try very hard through the fundamentals of and really cleaning up the nomenclature and definitions of the disease staging process. The goal is to take ambiguous and nebulous words like pre-manifesting, prodromal, and re-identify them and provide clear definitions that fit the biology of the disease. So we'll get into that. I actually put this here so it would remind me to make that point. So in Huntington's disease, the transition from not having clinical symptoms to having the clinical symptoms and then progression is protracted. The symptoms of Huntington's disease include cognitive decline, mood, behavior, and personality changes, involuntary movements, and speech difficulties. Importantly, there's no approved disease-modifying therapy, so the need is urgent and unmet. So this is a figure from the Tabrisi et al. Paper that was published in 2022, showing that the Huntington's disease integrated staging system spans the HD continuum and incorporates genetic, biomarker, and functional endpoints. So this is using the genetic diagnosis of the more than 40 repeats of CAG as the definer for stage 0. So just a quick genetics background. The exon 1 of the human Huntington gene is where there's a polymorphism in an expansion of the glutamine. There's normal variability in that expansion period, but below a certain threshold has no pathology or issue, and above that threshold leads to multinefarious effects. So this is using the cutoff of the CAG repeat within that exon 1 span that then translates to an unstable and deleterious protein as the determinant for stage 0. Importantly, there are no appreciable neurodegenerative changes yet for biomarkers, and certainly no clinical symptoms. When you start getting into stage 1, that is where there is a change, a shift in prognosis, because this is where the neurodegenerative process begins, which you can detect by measurable biomarkers. But importantly, no clinical symptoms are appreciated in stage 1. And we'll go into the specifics here in just a bit. For stage 2, this is when clinical signs are now manifesting. This can be observed by either motor or cognitive changes. And it's an important fundamental conceptual basis of the staging system is that it accumulates, those conditions accumulate across the disease spectrum. So we have the genetics initially plus the biomarkers. Now we're adding the clinical signs and symptoms. And these are usually detected by clinical reported outcomes or functional tests. And then stage 3 is where the effects of the individual's functional ability are manifest. So this is binned into mild, moderate, and severe. And for each of these different stages, there are different landmarks, which we'll walk through in just a moment. But the takeaway is that the HDISS is data-driven, biology-based, derived from extensive published evidence, and that, importantly, slowing progression across any of these stages would result in long-term decreases in morbidity and mortality. And we already lack a disease-modifying therapy for Huntington's disease, not for lack of trying. There are many programs and clinical studies that have been working at this for quite some time. But if we really do want to intervene early, and in the case of Huntington's with the genetic component, that opportunity is there. So this is what this is really driving at. There are several drug development companies that are looking to intervene earlier, and the staging system is designed to be a clinical trial enrichment tool to help that. So the basic approach that was used for the Huntington's disease integrated staging system, I want to completely acknowledge the heroic efforts of the entire Huntington's community to do this. This required a lot of work by clinician researchers, academic experts, CHDI, everyone coming together in the Huntington's Disease Regulatory Consortium at the Critical Path Institute, specifically under the Regulatory Science Forum. This was the body that really evaluated the first 3 points of this scale, of this methodology, specifically to agree on the questions that need to be systematically investigated, gather the evidence, and then debate and adjudicate what those landmarks are and which particular assessments should be considered. So the very wide net and the agnostic and unbiased approach that was applied early looked at many different assessments and then iteratively filtered that based on those assessments that were investigated in a larger population of individuals and that met the criteria for robustness and predictability and were put through the stress test of the algorithm development for prognostication. So it was a very complex process, and this group of experts were the ones who helped to evaluate that process. I'm not going to go into too much detail because I want to make sure I can go through each stage, but I mentioned the importance of genetics in Huntington's disease. Because it is autosomal dominant, I mentioned it has that expansion in exon 1. There's a threshold of what is deleterious and toxic, and that is depicted here, where you can see that expansions in lower number of expansions is not associated with disease. But as you move up into higher expansions, the penetrance is higher and the progression is faster and age of onset is earlier. So for purposes of the staging system, the particular threshold that was settled on was greater than 40 because that's where the penetrance is high and can be certainly consistent with the development of the disease. So now we're looking at the second stage of the staging system. So the first one was where we're looking at the stage 0 and that kind of, I don't know, like the reddish bar at the top. Now we're looking at the biomarkers. So clearly when you look at biomarkers, there's the opportunity for imaging biomarkers, there's opportunity for fluid biomarkers. For purposes of the staging system, the ones that we're focused on are volumetric brain imaging for two parts of the brain that are very important in the pathophysiology of Huntington's disease, the caudate and putamen, because it's the medium spiny neurons that are the most sensitive to the toxic effects of mutant Huntington. And so you can see through volumetric MRI reduced volume in the caudate and putamen. And this is a really linear effect where over the course of the different stages and years, you can see a significant decrease in the volume of both caudate and putamen. So this is showing, for purposes of the biomarkers, which again, recall that this is predating in terms of the disease continuum, clinical symptoms. So, but we know that these are brain areas that are important in Huntington's disease, so that it's tied to the underlying pathophysiology of the disease. Looking now at the stage 2, the blue area, looking at clinical manifestations. So this is, you've got the genetic predisposition, you've got the biomarkers, and then now we're looking at clinical assessments. There's two different ones that are being evaluated here. In the dark blue, sort of upward trajectory slope is the total motor score. And in the lighter blue is the symbol digit modalities test. Just want to explain why one's going up and one's going down. So in the total motor score, this is a component of a scoring system for Huntington's disease that looks at many different assessments. And the higher score is indicative of more disability or more accumulation of negative motor effects. In the case of the SDMT, that is a functional test. So losing that function and a reducing of the score is indication of worsening condition. And what you can see is that both of these assessments, whether the higher score showing disease progression or the reduced score showing disease progression, are certainly manifesting at stage 3. They're really not moving quickly. There's no steep slope to their trajectory in the early stages. So then looking now at the last aspect, functional capacity, we've got the cumulative insult physiologically of the genetic risk, the biomarkers, which are tracking with neurodegeneration, the signs, the clinical signs, and then obviously over time that will accumulate and lead to declines in function, which can be evaluated through assessment of activities of daily living. In this case, it was looking at total functional capacity and independence. So you can see again that these slopes are very flat in the early stages of the disease. There's not a lot of dynamic range or progression, and then the threshold effect, it's like falling off a cliff. So obviously, when you're looking at how an individual feels, functions, survives, this is clear, but it's for the families struggling with this disease, this is too late to start trying to intervene. We really need to take the opportunity to intervene earlier, and that's the goal of this whole process, to use the HDISS and the way that it's defining the progression across the full disease continuum to understand in the biology, the biological component of the underlying pathophysiology, so that we can better inform the right population, the right outcome measure, the right combination of the above in order to have more success with earlier interventions. So this is what's overlaid on this particular figure. All of these are from the Tabrisi et al. Paper. I certainly recommend that anyone wanting to delve into understanding the staging for their rare disease, really start with that as a scientific Bible. So just to summarize, the HDISS covers the continuum of Huntington's disease. This was developed by a multi-stakeholder group and the world experts for HD. It continues to iteratively evolve. This is a moving, it's a developing process, because we're getting more granularity and understanding for fluid biomarkers. We're getting better understanding and granularity. Some of these biomarkers are categorical initially, but more data can make them quantifiable. And so even though the HDISS is a very valuable and powerful tool in its current iteration, it is continuing to evolve. And this work that we're undertaking within HD Risk in the working groups, which are focused on biomarkers, clinical outcome assessments, digital health technologies, all of that will feed into further granular understanding of the staging system. And then once it was initially conceptualized as a research tool, but it's now being used actively for a clinical trial enrichment tool. So that's the potential. And as I mentioned, you can use this as a template for other disease areas. This is taking that same figure we've been looking at all along and kind of modifying it for the approach that we're taking in our critical path for therapeutics to the Ataxias Consortium, looking at spinal cerebellar ataxia and how we can try to understand the genetic component there, mutations in ataxin, which is also a CAG repeat disease, but has many different subtypes, and then understanding the opportunity for biomarkers and clinical signs within that paradigm. This can be, I don't want to make it imply that this is an easy transformation to take this to different diseases. It's certainly not a one-size-fits-all, but it is a very effective philosophical, conceptual framework that can be empowering. So I just want to, ask anyone, or maybe provide the challenge. If you're looking at applying the staging system, certainly make sure that you're asking the regulatory rigorous questions early. What are the concepts of interest? What's the best way to bring an outcome to the different individual stages so that you can have more success and rigor? And I want to thank everybody in the Huntington's Disease Regulatory Science Consortium. I mentioned it, it takes a village, it takes an army. This is my village and my army. So I want to acknowledge all the work that they've done to get to this, because I actually came over into this consortium in January of this year, and I just want to completely acknowledge the efforts that the consortium and the Huntington's community came together with to make this meaningful and successful. So I'll stop there, see if anyone has questions. Hi, Susan.
Hey, I'm just curious. So you'd said that more CAC repeats equals earlier onset, greater severity or faster moving. The stage 2 seemed remarkably consistent by age. So I guess my question is, to what extent do you think you can, if you've got fast progressing, first of all, fast versus slow, is there a big difference? It kind of looked like maybe there wasn't. But if there is, how can you use, we're in stage 1. Mm-hmm. Can you predict when stage 2 will begin? And the reason I'm asking that is from the perspective of access, reimbursement, HTAs.
No, these are really important questions, and there is, while there is the quote clean genetics in Huntington's disease for the penetrance and the autosomal dominant inheritance pattern, there's still heterogeneity. Someone can have the exact same number of CAG repeats and have a big difference in their age of onset and their rate of progression. What we didn't talk about in much of the work that's still being taken on by the consortium is looking at other factors that potentially could explain that heterogeneity, but also will help us have a better understanding of the underlying biology, like somatic expansion and other instability, rather, somatic instability. So there's just a lot more that needs to be understood. There's much more that is understood that I didn't have time to get into, so I don't want to make it seem as though this is all a black box that needs to be demystified. We do have a lot of really interesting work in that direction. The main Huntington's conference was in April this year, and there was a lot of really interesting work that was presented there. So it's an unclean answer to your question is that we do have some areas of maybe low-hanging fruit, but it's an active area of research to better understand it. And it's really that prognostication of converting within an individual and within stages and groups. If you can enrich for early stage 2, then you can start looking at that transition to stage 3 as you follow them over time. And so it's really important. Yes.
Thank you. That's a remarkably clean and concise way to look at disease staging. So congratulations, that was really very well done. So here's my question, and I apologize if I may have lost it as you went through this. How long did this process take from start to finish, and how many participants did you have?
Yes, this was a significant effort. I actually, because I was not at the helm, I know that this took several years to get from the initial conceptualization of how to approach it, setting up the committee for adjudication, discussion of landmarks. I think they started with something like 2,700 different assessments, and then filtered that down to ones that could be really trackable and were used in a larger population. So that I don't want to smooth over the complexity there, but I think this is what takes the community approach to really be able to bring the patient advocates, get the feedback from the individual's lived experience, get the clinicians, because really we have to get clinicians to overcome the uncomfortable reality that their patients are actually patients before they show up in their movement disorder clinic. So sometimes that's a hard change to get behind as well. So it took a large group a long time, but certainly worth the effort. All right. We actually have a showcase after, I think it's next. So if you have more questions about the staging system, please come to that showcase because we can dive into specifics there. Thank you.
Great, thank you so much, Tarina. As she mentioned, we are going to move into the showcases next. So great plug if you'd like to hear a little bit more about the Huntington's Disease Regulatory Science Consortium and the work that they're doing. You can follow Tarina as she makes her way up to the second floor, which is where that room is. If you're interested in the Polycystic Kidney Disease Outcomes Consortium and the work that they're doing, you can also follow Tarina or Soren, who's around here, also up to the second floor. If you would like to hear more about the resource that Lindsay Murray gave us a highlight of earlier, you can go right next door into this room and you can learn more about that. And then if you have an interest in Transplant Therapeutics Consortium and the work we're doing there, that is going to be in the London room, which is kind of behind the elevators that way. We will come back to this room around 5 o'clock for just a few short closing remarks, which will then be followed by the networking event, which will be next door a little bit after that, where we will have some light refreshments and hosted soda and water, and then a no-host bar. So we hope you stick around. Thank you. Hi, everyone. We're going to wrap up here just another minute or two. We're just going to have a few minutes of closing remarks before the networking event. So if anyone is in the hallway, wants to make their way into their seats, we'll just wrap up with a very short closing remarks on day one. While everyone is coming in and taking your seats, I'll go ahead and just give you a preview. So as soon as we wrap up with closing remarks, we'll have a few minutes, and then the networking event will start right out here in the hallway and in the room next door. As I mentioned, we do have some light refreshments, no host bar, and soda and water is complimentary, so we hope you'll join us for the networking event as soon as we wrap up today. Okay, so welcome back to Colin Hovingay. He's just going to wrap up the day with a few closing remarks.
I'll keep these short and sweet since the group is obviously a bit disseminated. I wanted to just say we have some great lessons learned from today and hearing from different stakeholders. Wonderful talks from the agency as well as the collaborations that within the ALS and Act for ALS community. We heard some wonderful things about endpoints and clinical trial simulation tools. And we closed the day with sort of tantalizing y'all with disease staging and some other discussions around endpoints and some of the new tools that CPATH has been working on. I want to pause here for a bit and just to say some thank yous for the people behind the scenes. Our PM and PC support and our EDs at CPATH have done just an amazing job putting this together. I want to just sort of give them a short hand of applause for them being here. In spite of all the challenges of getting here and all the orientations for people last minute needing to change their flights or going to trains and challenging schedules and getting slides last minute and trying to synthesize things, they've just really, all the way from Ubering from Pennsylvania, I mean, they've done a really excellent job of helping us pull this together, so thank you. The other thing I want to say, tomorrow morning we start is 8 o'clock. Yeah, 8 o'clock tomorrow morning we start. And tomorrow, it'll be more focused on RDCA-DAP and some of the initiatives there. We're having some discussions with patient-informed drug development. We have a session that is particularly on looking at some of the recent rare disease regulatory submissions and approvals, and really doing a little bit more discussion around from the pharma and regulatory experience in that space, both from CDER and CBER, to understanding what the total package and proof of evidence was, and just to try to give people some, compare and contrast. Without the rest of the day, I think Laura I forgot to mention, Health Canada and EMA will launch us in the morning, so we'll be talking with them initially, and they'll have a call coming in early in the morning to start our session off. So without further ado, I can adjourn for this evening, and hope everyone stays around for the networking and conversations that will instill following that. Thank you.